Hi there, my name is Debicki. I'm a clinical neurologist and an assistant professor specializing in neurocritical care. I'm representing our neurocritical care research group from Western University in London, Ontario, Canada. Today, I'm happy to talk about the use of functional near-infrared spectroscopy, or FNIRS, as a non-invasive assessment tool for detecting cognitive motor dissociation in acutely brain-injured patients in the intensive care unit. As we all know, some patients may remain unresponsive after a severe brain injury. The challenge is to determine which of these patients will recover from their brain injury and the extent of that recovery. It has been shown that some patients may have retained cognitive processing or elements of awareness despite being unresponsive to the clinical care team. This is thought to represent covert consciousness and has been termed cognitive motor dissociation. The hypothesis is that the presence of cognitive motor dissociation may be predictive of a favorable functional outcome and can be used as a biomarker for prognosis in the acute setting. The assessment for cognitive motor dissociation has involved advanced neuroimaging techniques such as functional magnetic resonance imaging, or fMRI, and electroencephalography, or EEG. These have been well established for improving the diagnosis in patients with chronic disorders of consciousness months or even years after their initial brain injury. The hope is that these or similar tools can be used in the acute brain injury setting to aid in early prognosis. But in order to do so, we need to better understand the relationship between neural responses to cognitive testing and functional outcomes. Our research group has been developing a battery of tests to assess the amount of covert awareness present in unresponsive patients. These tests range from resting state tasks to passive stimulation involving different types of somatosensory or auditory stimuli. It ultimately includes active command following tasks, including motor imagery, such as imagining playing tennis, and spatial navigation, such as imagining walking around your home. The brain response to these tests indicate just how much the brain is able to process and how aware the patient might actually be. So for example, if patients are able to show appropriate brain modulation during active command following tasks, it would suggest that the patient has awareness of their surroundings despite a lack of behavioral interaction and would be in keeping with cognitive motor dissociation. Although fMRI is thought to be a great way of assessing the brain's response to these tests, we have found some limitations to its use with patients in the ICU. This includes the risk of adverse events during the transport of patients to the fMRI scanner or during the scanning session itself, since the patient is not in the ICU. This often delays our ability to test patients until they are more stable. There is also the potential need for sedation to reduce agitation or movement during the scan. Of course, sedation will impact cognitive performance and limits the ability to interpret these tests accurately. To overcome these challenges, our group has developed testing paradigms using functional near-infrared spectroscopy, or FNIRS. FNIRS is an advanced imaging method that is often considered an optical equivalent to fMRI. Both of these techniques take advantage of the unique relationship between neural activity and changes in brain hemodynamics. Neural activity is associated with a change in the amount of oxygenated and deoxygenated blood in that brain area. This means that changes in oxygenated and deoxygenated blood in a specific area of the brain suggests that that part of the brain is active during a particular task. But instead of using a strong magnetic field like fMRI, FNIRS uses the properties of light to measure the amount of oxygenated and deoxygenated blood in different parts of the brain in relation to the different tests. Patients wear a cap with light sources that shine light on the brain and an array of light detectors to measure the way that that light interacts with the brain. It is a safe and a portable system where testing can be done at the bedside so that patients do not need to move out of the ICU. It also lends itself well to repeated testing on subsequent days. Our experimental protocol includes a resting state task where participants are asked to rest in the absence of external stimuli. It also includes a passive somatosensory task which involves repeated trains of electrical stimulation to the median nerve at the wrist, which is also known as an SSEP. It also includes an auditory perceptual task where different sounds, such as white noise, words that have been made up known as pseudo words, as well as a short story, are presented to participants in an interleaved manner. And finally, it includes an active mental imagery task where participants are asked to imagine playing tennis or imagine walking through their home. Today, I will show the results from a preliminary study involving a number of healthy control subjects that were used to validate the paradigm using NIRS, and from one of the ICU patients that was admitted to the intensive care unit with a stroke in a particular part of the brainstem called the pons. Testing with NIRS was done on day 22 of the patient's ICU stay, and at the time, the patient made some movements, but these did not seem purposeful, and there was no clear indication that the patient was able to understand instructions at the bedside by the clinical team. The most exciting results come from the mental imagery tasks, where participants were asked to imagine playing tennis. Healthy control participants showed changes in oxygenated and deoxygenated blood in specific brain regions in relation to the task. These regions are similar to those seen previously using fMRI techniques. Interestingly, our patient also showed changes to oxygenated and deoxygenated blood in similar brain regions. This suggests that our patient was able to modulate their brain activity in response to the mental imagery task in a way that was similar to that of healthy control participants. A similar finding was observed in the passive somatosensory and auditory perceptual tasks, where the brain regions responding to the different stimuli were similar in the patient as in the healthy control subjects. With the auditory tasks, similar brain regions were modulated when comparing the short story with the pseudo words and white noise, which suggests a higher order of language perception. Finally, our patient had similar patterns of resting state connectivity to that observed with our healthy control subjects in all of the major connectivity networks, including the sensory motor network, the auditory network, the frontal parietal control network, and the default mode network. Together, all of these findings suggest that our patient was able to modulate brain activity in a way that was similar to that of healthy controls. This is in keeping with the presence of cognitive motor dissociation and with the idea that our patient was able to understand instructions, but just not able to demonstrate that with physical interaction at the bedside. I really hope that you share our excitement around the possibilities associated with the use of NIRS as a tool for testing cognitive motor dissociation in patients with acute brain injury. It is a non-invasive bedside test, minimizing patient transport and the need for additional sedation. It has the ability to assess brain responses to different passive and active tasks and lends itself well to repeated testing over subsequent days. Finally, we might be able to use this test with other techniques, such as EEG, to complete multimodal assessments of patients in the ICU. Our research group will continue to refine our testing paradigms. There remains a lot of work behind the scenes to ensure that the way we measure and process the signals give us results that are reliable and reproducible. And we are currently testing more patients to determine the ability of this technique to predict functional clinical outcomes in our acutely brain injured patients. I'd really like to thank and recognize our incredible trainees, including Carnegie and Andrew, for putting together all of these results, to Sergio for working with the group to optimize our analysis pipeline, and to Matthew and Reza for taking on different aspects of adapting the testing paradigms and adapting the NIRS system itself for use in the ICU. I'd also like to acknowledge all the other members of the Owen Lab that ensure that this work is completed as well as it has been. This group is always a pleasure to work with, and we have generated so many interesting questions and solutions. I would also like to thank collaborators in the Neurocritical Care Research Group, especially Adrian Owen, Loretta Norton, and Tennille Gofton, as well as Keith and Rickson for their expertise in near-infrared spectroscopy and NIRS, as well, of course, as our funding sources. Thank you very much for listening.